Our topic for today is going to be about complex genetics. And this is really going to be a review of vocabulary and kind of some definitions. I'll explain a few examples, but this is mostly going to just be vocabulary review. So, one of the complex genetics topics we're going to cover is codominance. In codominance, there are two dominant alleles. That doesn't mean that there isn't a recessive allele. It just means that this trait comes with two dominant alleles. And when an organism inherits both dominant alleles, they're going to express both of them. Because the rule about dominance is that if you only have one copy of a dominant allele, you will express that dominant allele. That does not change. It's just now that we're expressing both dominant alleles. So an example here is with roan. Roan coloration is a combination of red and white hairs because both red and white coloration are dominant. An example in humans is blood types. A and B are both dominant alleles, and we still have a recessive allele in the form of O. A, you can either be homozygous for or heterozygous for. Same with B, or if you inherit both dominant alleles, you will be AB. And here's kind of what that means. So when we're looking at blood types, what you're talking about is basically a protein on the surface of a red blood cell. So here is antigen A, that's what we call these proteins, and it's shaped a specific way. You can see antigen B is shaped a different way. And then this is an AB person who has both antigens on the red blood cell. But the other thing about being a certain blood type is the antibodies you make. So an A person makes anti-B antibodies. And basically what that is it is an alert system. So an antibody will attach to a cell that has that specific shape, and it will alert your immune system that a foreign invader is present and it needs to come destroy that invader. So people with blood type A make anti-B antibodies. And vice versa, for people who are type B blood, they make anti-A antibodies. AB makes neither of those antibodies, whereas O makes both. Here's an example cross, so you can just kind of see how a Punnett square works with this. For blood types, we generally use the letter I as the base to represent the allele, and then we indicate what kind of dominant we have by putting a superscript. Polygenic traits are traits that are controlled by more than one gene. That means they have lots of genes controlling expression. And generally, you can see a polygenic trait because it has a wide range of phenotypes. Hair color, eye color, skin color, height, all of those are traits that have a wide range of phenotypes. There's lots of different expressions of height or eye color. Incomplete dominance. So in incomplete dominance, what we have is individuals that are homozygous looking different than individuals that are heterozygous. So red, these are snapdragons, and red is dominant. So we would expect to see capital R, when we look at their genotype. White is going to be represented by a different letter entirely because it's not going to be fully recessive. These are going to mix when we get down into our heterozygous individual. So when these are heterozygous, we generally represent them by having both the letter R and the letter W. And then we can get the original phenotypes back when we cross both heterozygous individuals. We can get a red, we can get 2 pink, and we can get 1 white. So here you can kind of see what that looks like. We use R and we use W. So this person would be pink, this person would be pink, this person would be pink, and that individual would be pink as well. So our next trait is sex-linked trait. Sex-linked traits are found on the X chromosome, and because of that, there's a difference in expression between males and females. So one of the biggest examples is color blindness. Color blindness is more commonly found in males because females have 2 copies of an X chromosome, like so, and they are less likely to get 2 copies of that recessive color blindness allele. Males, on the other hand, only get 1 copy of their X chromosome from their mom. So whatever is on that X chromosome, they have to express, whether it's hemophilia, like in the case of this Punnett square, or whether it's in the case of color blindness. Here's just another way to look at that, kind of more examples. Then we have X chromosome inactivation. In females, we have 2 X chromosomes, so we have to turn 1 off. This is usually random, and this is how we get calico cat expression. Calico cat, the black and orange are on the X chromosomes, and whichever one gets turned off, the other one gets expressed in that patch of fur. This is why you often hear people say that calico cats can only be female, because it requires 2 X chromosomes. You can have an XXY male calico cat, but then they're sterile. They won't be able to produce offspring. So this is kind of what you can see. There's the black allele, there's the orange allele, and whichever one gets turned off, that results in that patch of fur color. Epistasis. Epistasis are genes that will turn off the expression of others. So albinism is an epistatic gene. This hedgehog still has genetic information for hair color and eye color and skin color, but all of those have been switched off because he's expressing albinism instead. And then finally, environmental effects. This is sometimes referred to as epigenetics. The environment can affect expression in lots of different organisms. In sea turtles, the sex is determined by the temperature of the sand at hatching. They inherit the genetic information for both sets of reproductive organs, but only turn on a set based on the temperature when they hatch. Or Siamese coloration, this is my kitty. Siamese coloration is turned on based on which parts of the body cool off most after birth. And so that's where we're going to leave complex genetics. This is the last thing that we're going to talk about. Like I said, it's just kind of understanding all of these different terms and making sure that you understand how they could affect expression.